Hello friends, welcome to EPG Padshala. This module is called Bioprospecting. Bioprospecting is an umbrella term that refers ethical and sustainable utilization of the biodiversity of the planet Earth. As you know, the life on Earth has been evolved for the last 4 billion years and it's immensely diverse. That is the beauty of the planet Earth, its diversity. So how do we sustainably utilize these bioresources? Of course, the bioresources pays an immense utility for the humanity as a whole. As you know, probably more than 90% of the drugs that we use can be traceable to its origin on the plants as well as animals. So we have to use it sustainable and also in ethical manner. Bioprospecting is different from another term called biopiracy, which is exploitation rather than exploration. It's exploitation of traditional knowledge base, for example, tribal knowledge. So, biopiracy is unethical attribute, but bioprospecting is totally ethical as well as legitimate. So, in this module, we will see more about bioprospecting and how it differs from biopiracy, of course, and how the bioprospecting can generate funds that can fund, the conservation measures. As you know, most of the biodiversity on planet Earth is concentrated on the tropics. This is because of a theory called species energy relationship. As you know, the planet Earth, most of the energy from sunlight is falling on the tropics, and proportionately tropics are highly biodiverse, and as you see, biodiversity as well as the human population is positively correlated. So human population is also quite high, so demographic pressure is quite high, and naturally the countries in tropics are extremely poor. They are very resource poor, and most of these bioprospecting explorations were conducted by rich firms and rich investigators from non-tropic, subtropics and temperate regions. And bioprospecting can be related to or connected to the imperialism and politics as well. So we will see some of these intricacies of bioprospecting in today's module, as well as the legislation, CBD for example, as well as Kyoto Protocol and Nagoya Protocol. All these are connected with bioprospecting. So how does this protocols and legislations and international treaties affecting the bioprospecting? We will actually see that. And also finally we will see there is a tremendous current slowdown of the bioprospecting. And bioprospecting, again, it's not really cannot generate enough funds for sustainable utilization of the biodiversity. So why the reasons behind this current slowdown will be elaborated by the end of this module. So let's start this module. First, we will see some learning outcomes of this particular module. We should learn and definition as well as the concept of the bioprospecting, how this particular bioprospecting is defined. To know how sustainable bioprospecting can be an effective strategy to fund biological conservation initiative. To learn about various legislations and acts for ethical bioprospecting. And finally, to learn about the current challenges and future perspectives in bioprospecting. First is introduction. As you know, the life on Earth is 4 billion years old. So the 4 billion years of evolution by natural selection have produced a rich and complex tapestry of chemical constituents in living beings. A vast majority of biologically active natural products are impossible to be chemically synthesized. For example, mycotoxin. This particular toxin is obtained from marine fish, and as you see, this particular toxin has got lots of polycyclic, the cyclic structures as well as stereochemistry, which is almost impossible for chemical synthesis. So most of these complex natural bioproducts and biomolecules are not possible to synthesize chemically. So this particular resource is extremely important. More than 90% of the drugs that the currently world's market came directly from the organisms, especially from the plants. So it's extremely important, especially with reference to pharmaceutical sciences. Recent advances in recombinant DNA technology enables us to explore wild genes from the organisms and cloning them to further other organisms for various benefits. So again, genes is, genetic resources is also an important. It might not be the direct application, but indirectly these genes we can utilize from these organisms, and we can use these genes to clone it in yet another organism. One example that we will elaborate later is CRY gene, CRY gene, that is coding for the crystal protein. That is actually that is a biocontrol agent that we can use it in clone BT, BT brinjal or BT cotton GMOs. So bioprospecting is systematic and sustainable exploration of biochemical and genetic resources from nature and its further development into valuable products. So as we can see in this particular diagram, biodiversity can be utilized in many forms. For example, on the left side we will see a number of industrial natural products that could be industrial products, including anti-fouling, cosmetics, dyes, glues and other industrial products. And on the left side, the second one, you can see food and food additives, fodder, fertilizer, and below you can see biofuel, carbon capture and sequestration. So these energy uses also comes under the realm of bioprospecting. So it could be direct utilization of the biodiversity, or you can use a heuristic, the shortcut through the folk medicine or alternate medicine. For example, Ayurveda or Unani or Siddha or Chinese medicine, all these folk medicines. So we can screen the biological activity and animal testing, in vitro screening, and all standard protocol for the drug development. And finally, the drug development, that is a high value product. As you see, the drugs are extremely expensive. And biodiversity can also have a lot of cultural, aesthetic, and tourism and horticultural uses. And finally, biodiversity can be used for as a source of genes, for example, disease resistant genes or high yield varieties genes, that we can clone it into a suitable system and express and develop the product, that is the GMO. All this comes under the realm of bioprospecting. At the heart of bioprospecting comes ethics as well as the ownership. Many stakeholders would claim the resources as their indigenous, cultural, national and other privileges. The benefits arising, if any, would need to be shared in a fair manner, the so-called equitable benefit sharing. So that is extremely relevant component of this bioprospecting. Most of the biodiversity is centered around the tropics, which are economically poor. As I explained to you earlier, tropics are economically poor but extremely biodiverse. So the rich investigators as well as bioprospecting firms from temperate regions come to the tropics for exploration, which many anti-globalization ideologists term it as exploitation. Connection with imperialism or imperial colonialism, the coerced sample acquisition. As you see in those imperial days, the sample acquisition was coerced or forced. So this particular coerced sample acquisition is it's connected with imperialism, so it is not really an ethical manner of the bioprospecting. So exploration and exploitation, the terms have subtle difference in the meaning. Explorers and the downstream bioprospecting industries from the Europe had until very recently been in fact exploiting the tropical countries with virtually no benefit sharing. For example, when the Britishers came and colonized India, a number of plant resources from Himalayas as well as mainland India had been transported. And once you can see, when you go to Kew Garden, for example, I've been to Kew Garden 2 times, so you can see a number of Indian plants already cultivated in Kew Gardens in London. So this kind of exploitation of the natural resource through imperialism is, it's one of the major attribution of biopiracy as well. Instead of randomly screening the natural products, which is very slow and expensive, One estimate say you need to screen 250,000 extracts for just to develop one drug candidate. Industries take the heuristic, that it's a shortcut, screen extracts from traditional folk ethnomedicinal remedies. So biopiracy is a term that used to implicate unethical sample acquisition, especially through exploiting the tribal knowledge base. One example is the development of anti-cancer drugs vincristine and vinblastine. Vinca alkaloids from the rosy periwinkle, it's a plant native to Madagascar, Africa, and part of the folk medicine for thousands of years. So these firms, the anti-cancer drugs were developed by the American firms with no benefit sharing with Madagascar. When traditional knowledge is exploited without an informed consent or benefit sharing, the exploitation is termed biopiracy. So bioprospecting had been regarded as a fund generator, a sustainable fund generator to fund conservation efforts of the poor countries. So many countries in the tropics are economically underprivileged to fund the biodiversity conservation programs. Bioprospectors from the developed countries negotiate per sample cost for sample acquisition from tropical countries. Part of this revenue can be channeled for biodiversity conservation programs. So there are several legal and government legislations for bioprospecting. Perhaps the most important and most famous one is in 1992 Earth Summit in Rio de Janeiro in Brazil, which actually ratified a protocol called Convention on Biological Diversity, or CBD, ratified in 1992 by 196 countries. So it is the first legal framework for the bioprospecting. So CBD enabled, made it compulsory to get prior informed consent, or PIC. So it's mandatory for sample acquisition from anywhere in the world. So PIC needs to be obtained by the sample, sample acquisitor or bioprospecting firms from the sample provider. Also made agreements for technology transfer, impact assessment, that is environmental impact assessment, technical as well as scientific. In 2010, Nagoya Protocol in Japan had been established. So this is the legal international framework for the transparent, equitable benefit sharing. So the benefit sharing, if any, arised out of the bioprospecting, needs to be shared between all parties in a fair and equitable manner. The protocol entered into force in 2014 and had been ratified by 96 parties, including the European Union till date, while notable exceptions include United States, Canada, and Russia. What were the consequences of the CBD and Nagoya Protocol? There were pros and cons as well. Pros is the good attributes are dramatically reduce the incidences of illegal sample acquisitions. Such cases of suspected biopiracy could be tried at International Court of Justice in Hague. While cons are extreme bureaucratic hurdles with sample acquisition. Most of the bioprospectors lost interest in bioprospecting from the tropics. They now leverage combinatorial chemistry-based approaches because the sample acquisition has become tremendously bureaucratic process with multi-step bureaucratic hurdles. So sample acquisition is no more become viable or no more become an easy task, which forced a number of countries, the bioprospectors, to move away from bioprospecting itself. What is the scope of bioprospecting? Value of bioprospecting as well as biodiversity is relative as well as ambiguous, so no consensus exists. The estimate ranges between $21 per hectare of land to $9,177 per hectare. So this disparity, huge range, is partly attributable to where you do this bioprospecting. For example, if you do that in the tropical region, especially in places where endemism is extreme, for example, biodiversity hotspots, so the value is relatively higher than a temperate region. So that is the reason for that. So $9,177 per sample is when you do the acquisition in the hotspots. So $100 per sample promised by the GlaxoSmithKline, it's a US firm, to the Brazil, but of course that later the deal was cancelled again due to many factors, especially with the aftermath of Nagoya Protocol. Drugs are most prominent high-value product in bioprospecting missions. So because the drugs are quite expensive, especially chemotherapeutics, for example, paclitaxel, this is an anti-cancer reagent. So the drugs are the de facto, product of the bioprospecting. Drug development is a very rigorous process. Efficiency of the candidate drugs have to be unambiguously proved through rigorous statistics-backed scientific methodology. So it's not that easy for developing a drug, commercially viable drug. To overcome these obstacles, the herbal formulations are often developed into polymolecular botanical supplements, functional foods, and nutraceuticals. One example is BGR34, a purportedly anti-diabetic formulations developed by CSIR India, that is developed by National Botanical Research Lab, NBRI in Lucknow, that BGR34, which they claim to be an anti-diabetic natural supplement. Because most of the firms take this route, because to develop a scientifically evidence-based drug, it's quite a challenging task. So lucky finds, the term lucky finds refers, when you do this screening of natural products, trial and error method for screening large number of natural products to get one lucky find, so one successful drug candidates. Random screening of natural product is immensely slow and expensive venture, but sometimes rewarding. So as explained to you earlier, you need to screen almost 250,000 candidates for just to get one possible drug candidate. So one famous example is the United States National Cancer Institute's large-scale natural product screening for anti-cancer activities. The program screened extracts of over 70,000 plant species over the course of around 2 decades and resulted in the development of just one drug, paclitaxel, so that is a taxol derivative, which is a commercial successful and chemotherapeutic agent, which is a blockbuster drug. The blockbuster drug means the profit generated is quite high. Another example is cyclosporin A, which is an immunosuppressant. The story is very interesting that we will elaborate in the biopiracy module. A Swiss researcher on a family vacation to Norway brought back the soil samples, and from the soil sample he got some fungus and developed the drug from the soil fungi. So this drug cyclosporin A is it's a lucky find. Cerulomycin A is an immunosuppressant as well as an antibiotic. So it is developed by Dr. Javed Agriwala from IMTAC, CSIR IMTAC, Chandigarh. So Javed Agriwala is a Bhatnagar awardee as well. So he brought back some soil samples from Spiti Valley, Lahul Spiti in Himachal Pradesh, in the Himalaya valleys, and he developed this compound. So again, another example of the lucky find. So it is quite rare, but again, it's promising. Bioprospecting is there are a number of biases and cognitive biases in this bioprospecting. Some of these biases, let us see. One is that bioprospecting explorations are mostly targeted to the land masses. However, more than 70 or 71% of the Earth's surface is ocean. So only very less explorations to the ocean, although the ocean covers most of the Earth. At present, the world has only 5 manned submersibles suitable for the deep sea diving for more than 3,000 meters, a major hurdle for the bioprospecting of the world's oceans. So one aphorism says that humanity knows the moon much better than we know of our oceans. So this bias is very prominent across all disciplines. So bioprospecting of the open sea is also extremely rare. An estimate suggests that 99% of all ocean explorations of bioprospecting happens within territorial waters. So territorial waters means those coastal region within 12 nautical miles, or 22.5 kilometers from the coastline. So beyond 22.5, not much of the explorations happened. So only 1% happened in the deep seas. Almost 99% of these explorations are coastal regions. So most of the sea remain to be explored. Only a very few firms target the marine biodiversity. For example, Diversa Corporation, Magellan Bioscience in the US, and Bank in Norway. So it's extremely rare for the firms to target the marine bioresources. Of course, there are success stories in marine bioprospecting as well. For example, ultra-thin is an enzyme from the genes of archaebacteria living in the Pacific hydrothermal vents. As you see, these are benthic habitats which is highly specious, the hydrothermal vents. Another example is exosap IT, which is from the ocean shrimp. It's an enzyme, SAP, that means shrimp alkaline phosphatase. SAP exosap IT is used in PCR reaction cleanup before the DNA sequencing. So another related cold tolerant enzyme is called Antarctic phosphatase from the Antarctic bacteria. So all these are success stories from the marine bioprospecting. Some other examples include selenoporamide A from marine bacterium Selenospora, protease inhibitor and anticancer compound. Another example is actinicidin 743, that is, it's a part of trabectedin. It's an approved anticancer drug developed from marine tunicate sea squid, Tenacida turbinate. Another example is enzyme luminase, which is pulp bleaching enzyme, originally collected from the genes of geothermal sediment samples from Kamchatka Peninsula, Russia. At present, what is the current status of the bioprospecting? Bioprospecting has come to a dramatic slowdown for the last 2 decades. So one main reason is the bureaucratic hurdles, post-enforcement of various international agreements, especially CBD, post Rio de Janeiro Earth Summit of 1992, as well as Nagoya Protocol. It has increasingly become an impossible affair to acquire even a single sample from certain countries, for example, Kenya, that forbids all bioprospecting activities for the foreign companies. Another reason is the bioprospecting being extremely slow process that demand a gargantuan effort, which is labor intensive. Most of the world's traditional medicines are now known to the scientists. Although straightforward and heuristical advantageous option have now become saturated. So it's no more viable option, to look for potential candidates from the traditional or herbal medicine or indigenous knowledge base. It's now becoming more and more clear that the bioprospecting is highly unlikely to become an economic driver to support the biodiversity conservation measures by the poor countries. So the biodiversity conservation measures are, of course, you need to generate a lot of fund, but to generate the fund, as previously thought, bioprospecting is no more an economically viable option. Drug discovery companies these days increasingly rely on the combinatorial chemistry opposed on natural products for the source of new drugs. The so-called total synthetic attribute is now being followed by the drug companies rather than going for the bioprospecting. So this step is something called molecular docking. It is a two-step approach for the drug development. So instead of screening, firms identify and elucidate structures of the natural products and construct a searchable database. Some examples of the searchable database include Inter-Bioscreen Library, Supernatural Library, and so on. These databases can be searched to find potential structural matches. For example, an inhibitor molecule that perfectly bind with three-dimensional structure of an enzyme that need to be targeted for the biological pathways, a process known as virtual or in silico screening through the molecular docking. So in summary, biodiversity prospecting or bioprospecting is an umbrella term that's used to refer the sustainable utilization of the biodiversity for the betterment of the human life. Two of the most important international legislations that govern sustainable biodiversity worldwide are CBD of the United Nations Convention of Biological Diversity and Nagoya Protocol. These agreements have intended as a legal framework for equitable benefit sharing and prior informed consent for resource provider. So a major bias of bioprospecting is that most of the bioprospecting firms now exploit only the land masses, and virtually the world is now like 71% of the Earth's surface is water. So it's covered by the ocean. So bioprospecting firms exploiting the oceanic resources are extremely less. So that is actually a very, very important bias in the bioprospecting. So it has been classically projected as an a sustainable fundraiser for conservation measures. So because most of the species rich countries are tropical and they are economically less privileged, you need to generate a lot of funds for the bioprospecting as well as for the conservation. But nowadays it has become more and more clear, increasingly clear, that the bioprospecting is no more a viable option for fundraising because it involves a lot of bureaucratic hurdles. So nowadays most of the bioprospecting firms, especially pharmaceutical firms, are taking an alternate course through combinatorial chemistry. So in summary, bioprospecting is an ethical as well as legally binding sustainable utilization of biodiversity of the planet Earth. So biodiversity is extremely important, and the value of biodiversity is more than clear for the humanity. And most of these biomolecules are extremely complex. So it is impossible for a total synthetic approach. It's not possible to generate these kind of compounds. So you need to extract these compounds from the nature through bioprospecting. Though these rules and regulations have now become increasing clear that it has actually impeded the flow of information because the sample acquisition has become extremely tedious process. Thank you.